Hello and welcome. Before we get started today, a note to all of our participants that your microphones are muted and your cameras are turned off, so there's no need to worry about any background noise on your side. Let's first do a quick sound check for all of our listeners, and we'll also wait for people to join as they come on board the call. Please write in the chat where you're attending from today, so we can make sure that you can hear me. Okay. Okay, lots of Midwesterners. We have a colleague from Mexico and someone from Germany. That's great. So sound is working. That's great. Well, we have just over 50 registrants today. So let's wait till they join. We only have about 18 right now on the call. Chicago, my hometown. Love that. Okay, I think we can go ahead and get started. And everyone can just, jump in as they join the call. So welcome to our thought leadership webinar, Changes and Directions in Applying Model-Based Drug Discovery and Development. This is the first in a series sponsored by Research Solutions. We're a leading provider of workflows that streamline how organizations discover, acquire, and manage scientific articles and data. My name is Sharon Madden-Boudicca, and I've been the Director of Content Management at Research Solutions for 6 years now. Prior to this role, I worked as a publisher for nearly 2 decades, concentrating on building out distribution channels of original published research. Today, we partner with Wiley, the world's largest society publisher, serving various disciplines and research communities, and the American Society for Clinical Pharmacology and Therapeutics. They're an organization founded in 1900 whose mission is advancing clinical pharmacology, translational science, and therapeutics for the benefit of patients and society. The ASCTP is the largest scientific and professional organization serving the disciplines of clinical pharmacology and translational medicine. Together today, we'll explore model-based, also known as model-informed drug development and discovery methods, their advancements, the knowledge it generates, as well as the decisions that they facilitate. On behalf of Wiley, the American Society for Clinical Pharmacology and Therapeutics and Research Solutions, thank you so much for joining us today. At this 1-hour event, I welcome the distinguished panelists. Oh, let me just give a quick, oh, we're back to the slide. Sorry about that. Okay, we welcome one of our panelists, Vikram Sinha, was unexpectedly detained, so he may join us later in the call. So I'll introduce him once he joins, if he's able to join us. He had a delay. So I welcome Priya Jayachandram. She has a Doctor of Pharmacy. She holds a Master of Science in Engineering and is an Associate Director and Pharmacometrician in the Clinical Pharmacology and Pharmacometrics Group at Pfizer. Priya is also an editor in training for the journal CPT Pharmacometrics and Systems Pharmacology, which is the official journal for the society ASCPT, as well as, I'm informed, the International Society for Pharmacometrics. So welcome and thank you for joining us today, Priya. Our own Robert Radloff holds an MBA and studied technical biology in Germany and Australia with a focus on clinical pharmacology and bioinformatics. Robert has over a decade of experience in the pharmaceutical and medical device industry and has contributed to several publications in the clinical and regulatory research domain. As the Director of Product Development at Research Solution, Robert is a strong supporter of the open innovation approach and curious about digitization and artificial intelligence solutions. Robert, glad to have you with us today. I also just want to mention a little disclaimer. All the panelists represent their own views during our discussion today, and not necessarily that of the companies for which they're employed or the societies to which they belong. So let's go ahead and start with a poll to gauge audience knowledge of model-informed drug discovery and development, which is also been referred to as MID3. The 3 Ds stand for drug, discovery, and development. So here's our first question. What's your current level of understanding of model-informed drug discovery and development? Give everybody just a few minutes to answer. Could be your first exposure to the topic or you use it every day. Just let us know so we can gauge audience knowledge level. Give everybody a few more minutes to enter their. Let's ask Gabi, who's driving the polls. Do we have almost everybody having responded? Do you want to close it out in about half a minute? Okay, let's see the results. Okay, so for quite a few of you, over half of you, this is your first exposure to the topic. So, and about 25% use it in your day-to-day work, and about the same percentage rely on it for outcomes and decision-making. So this is very interesting. So we have half our audience, it's brand new, and the other half really utilize it every day. So this is very interesting. We have one more poll to ask you all. Gabi, if you could service the poll. What's your main challenge with regard to improving research modeling? It can be one or it could be all of them. Okay, Gabi, do you think we've given enough people to time to answer the challenges poll? Have you gotten enough responses? Okay, so we've got about 2/3 say that one of the challenges is their own awareness or their immediate team's awareness. The ability to assess research outcome to affect change, about 1/3. And then another 1/3 varying views of utility of differing approaches or methods, and organizational priority seems to be less of the problem. So this is very insightful. Priya and Robert, I'm sure you find that to be good in terms of identifying the audience that you're speaking to right now. So very interesting. Thank you very much for responding to the poll, everybody. So to launch the discussion, we viewed a paper published in the journal Pharmacometrics and Systems Pharmacology, and which is a cross-disciplinary journal published by Wiley. And it's devoted to publishing advances in quantitative methods as applied in pharmacology, physiology, and therapeutics. The journal applies a systematic approach to understanding how drugs work, how much knowledge this understanding generates, and the decisions that it facilitates. It can also reflect the state of science in this area. Specifically, the journal publishes primarily in the areas of pharmacogenetics, modeling and simulation as applied to the design and evaluation of clinical trials, but it also explores the areas of systems pharmacology modeling, disease modeling, computation pharmacology, bioinformatics, comparative efficacy, and cost effectiveness. And I believe that in the chat, Julia posted a link to the article, which is an open access article. If you haven't had a chance to have a look at it, it gives the overview of model-informed drug discovery and development, which is the basis of our discussion today. So MID3 has proven to play a major role in the efficient delivery of new therapies by both increasing the confidence in decision-making across drug development, as well as by eliminating costs or reducing cycle times. So Robert, we have a slide about data inputs here. Can you tell us a bit about the research inputs that go into MID3?
Yeah, sure, Sharon. So thanks for the invite. And yeah, welcome all today. So hopefully you hear me loud and clear. So that's good. And my German accent, so sorry for that. Anyway, so I would like to start with the basics, because I think my last touch point with the in vitro or in silico drug development was 10 years ago in the university in my research thesis. So that's why I'm pretty glad to refresh my knowledge here again. So with like experts like Priya, so that's very welcome from my side. So lifelong learning, so something like this. Yeah. But anyway, so the data inputs for model-informed drug development, or also model-informed drug discovery and development, so MID3. So mostly these are pharmacokinetics experiments taken from the lab. So that is the data coming out of in vitro experiments, and also from clinical trials where we are doing in vivo patient pharmacokinetics. So these are mostly in most clinical trials, I think phase 1 and 2 and also 3. So, and later on in the real world trials, we get less experience, I think, with the pharmacokinetics. But this is one of the major challenge we get in the phase 1 clinical trials, where we have limited of patients involved in that. And that's why it's really welcome if we have later on in silico, so computer modeling with these data from a limited patient set. So that's the basic idea here for pharmacokinetics. And I explain in the next slide then also what pharmacokinetics is and pharmacodynamics, because I saw these 54% have also basic understanding like me in this process of model-informed drug development. So that's why, yeah, feel very welcome, so that you also get something basic from my point of view. And then the pharmacodynamics. So this is all about drug-drug interactions, because in the clinical trials, you have a really homogeneous set of patients. So you get not a lot of exposure to other drugs and other substance which can affect the drug efficiency or efficacy. Yeah? So that is something we should also be aware. We cannot expose the patient with a lot of different drugs in those trials. So because it should be like a lab trial. Yeah? So that is so there's a protocol, and that's why the drug exposure is limited to other drugs, which can affect later on in the real world those side effects. Yeah, but I think Priya can also tell us something about that later. Yeah, and then the main concept of pharmacokinetics is also the absorption of the drug, but this is also in the next slide, so I do not want to advance that. And
yeah,
other
Bobby, maybe you could advance to the next slide. That would be great. Thank you.
Yeah, thank you. All right, and then we have those principle of pharmacokinetics, as I said. So this is, I think, a nice picture from the EU PATI. So they are explaining to patients what is pharmacokinetics, but if you trial patients, so then you can see, oh, what's that pharmacokinetics? I do not want that. Yeah, so that is also in some clinical trials for pharmaceutical drugs. You can volunteer to be in the pharmacokinetics and pharmacodynamics subset. So that's why it's good to explain to patients what they are doing with your data, and that you are also delivering something for the global community of your population. So that is also good that physicians and clinical researchers can explain this topic to the patients. So basically, it's about the ADME acronym. So that's the absorption. So you take in your medicine, which can be in various formulations, like here a capsule. And then the distribution, so a distribution all over the body, so systematic approach, or even if it's a targeted approach. So maybe you have an infection in the knee, so you do not want to get ruin your gut microbiome. So that's why it's good if it's very targeted to the site of your disease, I would say. The distribution, so that was the distribution, and the metabolism is then so breaking down the drug into the active ingredient so that they can take effect in your body. So that's the metabolism, which is hugely done in the liver, but also in the kidneys. So that is the main route of excretion, where we go then in the last step. So how does it leave our body? So this is called the ADME process, I would say. So that is then it gets out of your body and hopefully without any side effects, but there are no side effects. So without less side effects, I would say. Yeah, so this is basically the concept of pharmacokinetics is, when do you get the drug and how is the drug absorbed and distributed and excreted. So anything else, Priya, maybe I missed something. So then please add something if you want to say.
No, I think that was great, Robert, actually. Thank you for giving that introduction about ADME. I think, I often think about PK in terms of what sort of effect that drug is having on the body. And I think that's basically what's being described here really well. So nothing really to add.
Good, thanks. So then we can go to the next slide if there are no. So I think the audience is also welcome to type questions in the chat. I'm not sure, Julia, if you want that. So that's something I think we did not explain before. So the audience can also write any questions in the chat so that we can also take them on the fly or later when we finish that. But anyway, so the next concept is, I think that's a little bit advanced now, so it can be explained in various steps. So this, I do not want to explain to patients anymore. So this is more for clinical researchers and scientific community. But I think it explains nicely that the pharmacokinetics and pharmacodynamics are interacting with each other, That's also a bad word for Germans. So first, you have a drug dosing. Yeah, so that is here maybe a tablet. I cannot see that really good. And in this tablet are the drug molecules. So that's called the formulation. And then in the end, so now we are going to the ADME process. The first one was the absorption. So you get this inside the body. And if you see the curve, so this is a drug concentration time curve. You see if you get a tablet, then it's another concentration time curve. Then you get it intravenous in your body via a intravenous application. So that is the pharmacokinetics. So how does the drug or the active ingredient later on interact within your body and where? And then the next one would be the site of interaction. So we are going into the pharmacodynamics. So where's the target? So the drug should then reach his or her, it's a target. So then we have an effect, so a response, hopefully. So then, this is also distributed over time. So maybe a good example would be aspirin. This is taking an effect, I think, after maybe 30 minutes. So you will see the highest response after 30 minutes, and then it will go slowly down, and then you need hopefully not another aspirin, or maybe that's the brand name that's not good. So maybe this also, but I'm not sure. So that we are not making any advertisement here for aspirin. So maybe paracetamol is also another example. So yeah, this is the effect time curve. So that is more or less pharmacodynamics. And then in the end, we have also the effect and drug concentration time curve. So these are all maybe mathematical models, so which you can then in the end model in our our model-informed drug development, I would say in this case. So this is exactly what we are taking out from patients or from laboratory assays, in vitro experiments, and then modeling it to maybe other population, because it's also a hot topic. You cannot do many clinical trials for pediatrics, newborns, because most of the children do not want to attend trials, or maybe the parents do not want that they attend a trial, so they want to be a guinea pig for their children. So that's why I think it's also good you can advance this model also to pediatrics, where we have only little patient population in clinical trials. So that is also a good use case. Maybe we can explain later a little bit more on that. But yeah, so this would be a good use case for model-informed drug development. All right, so that's all I have to say.
thank you for explaining the process of how drugs are absorbed into the body and
Sure.
all of the side effects and everything associated with drug absorption. Now, Priya, can you help us understand why MID3 matters in drug development?
Yeah, certainly, Sharon. And thanks for having me here. I'm very happy to be here today. So the way that I like to think about MID3 is that it's essentially a series of mathematical models like pharmacometrics, physiologically based pharmacokinetics or PBPK, and quantitative systems pharmacology or QSP modeling. And they come together to integrate information on a drug, a disease, and a mechanism level. And if we can advance to the next slide, we can actually take a look at how these models come together with the different sources of input that Robert was talking about. So pharmacometrics models, they're really used to characterize the drug pharmacology, as Robert just explained, the absorption, distribution, metabolism, and excretion. And they're comprised of population PK models. And population PK models are really characterizing that concentration time profile or the exposure of the drug. And we also have population PK/PD models, and those are essentially characterizing the exposure and response relationship. So characterizing the rate and extent of exposure of the drug, and then how the body is responding to that drug. And so in that way, pharmacometrics models are really characterizing the drug pharmacology and response. Next, we have PBPK models. These are models that use information on both the drug and the physiology level. And so they consist of a larger number of compartments than pharmacometrics models usually contain. And those compartments correspond to different organs or tissues in the body. And so we can develop a quantitative mechanistic framework that can be used to predict the plasma and tissue concentration time profiles. So an example of this would be, for example, if we wanted to extrapolate a dose in healthy volunteers to one disease population, we can do so if we have the relevant physiological properties of that disease population. And then we have QSP modeling, and this really expands on the concepts of PBPK modeling to construct a deeper understanding of the disease pathology and its response to a drug. So with QSP modeling, we can examine the interactions that exist between the experimental data from drug studies and the biological processes that are related to the disease. And those biological processes can be pathophysiology, they can be specific disease pathways like a signal transduction pathway, or other things. And so what we do really with QSP modeling is integrate that regulation and metabolic level of information about biological pathways with drug mechanisms to really advance our understanding of how drugs affect disease. And there are several uses of QSP models, particularly in the drug discovery and preclinical development arena. But they can also be used to provide information for downstream pharmacometrics models. So for example, we might be able to identify determinants of exposure by looking at the pharmacogenomics, maybe identifying that there are rapid and slow metabolizers. That is useful for downstream pharmacometrics models to tailor dosing based off of the pharmacogenomics. We can also improve, in general, our translational understanding of PK/PD responses across species to enable us to have more model-informed predictions of clinical outcomes from those preclinical models. And that, in the grand scheme of things, really speeds up drug development for us. And so in this way, QSP modeling really helps with hypothesis generation to guide the development of study designs and future experiments and to save time in the drug development space. And as Robert already mentioned, the inputs to all of these different model types, they come from preclinical experiments as well as first in human studies, phase 2, phase 3, and even some post-marketing studies.
Thank you, Priya. What are some of the current challenges in applying MID3 to drug development and regulatory decision-making?
Sure. So I think some of the challenges in applying MID3 relate to acceptance. I do think that acceptance of MID3 has previously been a challenge, but it's increased significantly in recent years. I think publication of successful efforts of MID3 has certainly contributed to that acceptance. CPT Pharmacometrics Syst Pharmacology, the journal to which you referred to in the beginning, which is a part of the American Society of Clinical Pharmacology and Therapeutics and the International Society of Pharmacometrics, is really a flagship journal in the field that has published several of these examples. But what I do think is a challenge still in terms of acceptance is communication across disciplines. So for example, the communication between pharmacometrics and statistics. And PSP, for example, has really tried to address this issue by bringing forward special issues that are bringing together multiple disciplines. And I hope we'll see much more of that in the future. Another challenge that I think exists within MID3 is the implementation of these frameworks uniformly across therapeutic areas. So one of those challenges that we face right now is with newly evolving modalities like gene therapy and mRNA-based therapeutics and gene editing. Rare disease is a therapeutic area that has seen an exponential increase in these modalities, but it's really difficult for us to be able to apply conventional, for example, drug dosing principles to select a dose when gene therapies require consideration of cell type, cell viability, viral vector concentrations, transgene expression, et cetera. So all of a sudden, the traditional questions that we have and approaches that we have are sort of being challenged by some of these new therapeutic areas or new therapeutic modalities that are existing in specific therapeutic areas.
Well, this
and then I also think,
I'm sorry, Priya, I just have a question about, does this, especially in new areas that we don't have a lot of protocol for, let's say, does MID3 affect regulation, or does it improve it, or conversely, does regulation govern MID3? Like what's the driving factor there?
I think it goes both ways, actually. I think, one element is when developers work on essentially building these models in order to be able to quantify and answer certain regulatory questions, there's a body of evidence that grows that sort of speaks to how these approaches can be used. And then other developers can utilize those approaches and expand on those approaches. But then also, when those approaches are analyzed on a larger scale, they can be used to inform guidance development for a new therapeutic area, for example, or a new therapeutic modality. And so all of a sudden, the converse is happening. It's no longer just, are we simply developing new methods and methodologies, but we're also developing best practices to guide how we're going to analyze data from these types of therapeutic modalities going forward. So I think it's a two-way process for sure.
Well, that's very interesting. So what are some of the advancements in model-informed drug discovery and development that we can expect to see in the future?
Sure.
Do you have any insights on that?
Yeah, I'd be happy to share some insights on that. So I think, one of the areas that I see integration of modeling approaches is in core areas as well as emerging areas. So some of the core areas where we have integration and we need more integration of is, for example, QSP and pharmacometrics modeling to support end-to-end applications of modeling from drug discovery all the way through post-marketing. And also just in general to advance our understanding of the physiological basis of specific diseases and how they're affected by a drug. But also, we will hopefully be seeing a lot more integrated modeling in emerging areas. For example, pharmacoeconomics and pharmacometrics models in order to sort of address the gaps in clinical evidence that can be used to inform reimbursement decisions. And also machine learning to probe the analysis of really large databases of information like healthcare databases and unstructured resource databases like real world data from registries, et cetera, in order for us to learn how to use all that information to inform essentially the totality of evidence that we have to bring a drug to market. And along those lines, I also think that hopefully we will see more advancements in open source software and curated databases that house a lot of this information in order for us to be able to promote collaboration and usability of that data. And I already spoke to communication across disciplines, but I think that's another area that we'll hopefully see more advancement in, and that will help with acceptance of MID3 and its impact across the field. And personally, an area that's really important and interesting to me is the increased training that I hope we'll be able to see in the quantitative sciences and how that can hopefully be increased with regards to geographic diversity. And what I mean by that is we need to strengthen our global initiatives by building partnerships and learning between, for example, the EMA, African Medicines Agency, FDA, PMDA, in order to share and evolve those best practices, because only then can we really move forward to new heights within MID3. So hopefully in all of those different areas, we'll have some advancements.
Okay, well, tell me a bit then about the journal. I mean, in what ways does the journal Pharmacometrics and Systems Pharmacology serve the needs of the ASCPT and the International Society of Pharmacometrics. I mean, how can you leverage the journal to reach those goals?
Absolutely. So I think, you know, PSP is a journal that essentially highlights examples and applications of successful modeling approaches in drug development. And it covers the core areas of QSP, PBPK, and pharmacometrics, but also emerging areas like machine learning and artificial intelligence. And in the future, hopefully, an integration of those core and emerging areas together. What the journal does also have is, in addition to sort of the original research articles that support all of those different modeling approaches that I just described, it also has a number of tutorials, and those tutorials are instrumental in teaching the next generation of quantitative clinical pharmacologists and thought leaders about how to use these techniques in drug development. I know I myself, when I was learning at the very, very beginning, and I still am always learning, as Robert said, we're perpetual learners, I rely on these tutorials, and they're so incredibly useful because they're put together by thought leaders and experts in the field, and they really help to guide our knowledge and our growth in the field. And also what PSP does is it's expanding its scope to now include preclinical translational applications. And so we're really spanning the full drug development pipeline to be able to see how we can utilize all of that information and showcase examples of utilizing all that information in order to be able to bring drugs to market. And one of my favorite aspects, I think, about PSP is the promotion for sharing model codes, because that really, really helps those that are working in a very, time-constrained manner to be able to utilize already existing models in order to be able to reuse them, to adapt them, to apply them to new applications. It saves a lot of time and also helps to sort of streamline the applications that we have.
I see a question in the Q&A. It was about the subscription to the journal, and it is an open access journal, so there is no subscription required. So yeah. And earlier, they're asking about impact factor as well. The impact factor, I looked it up real quick. Last year, the impact factor was 4.339, 2021. So that's a healthy number. Yeah.
Yes.
So very good. So it sounds like that you're not only reaching out to society members, but also to the broader research community to be a resource on this specific type of information. So let me turn to Robert for a second. Robert, can you talk a little bit about the curation of data in your own experience? How do you leverage data sets and literature to advance your own research? I mean, do you rely on MID3 or other processes or?
Yeah, so from my research background, so I can just not talk about pharmaceutical industry because that is highly regulated. So they have other standards than at the university or research institutes. So at least in our lab, which was beside a hospital, a big hospital in Germany. So they had their own standards, lab standards, but in the end, so there's at least for the pharmaceutical industry, I think I need to look it up, this clinical data interchange standard consortium, which applies some standard way of data standards for drug development. So it's a good, I think a good setup if a drug development community use the same standards, so they can also interchange the models and the data which is coming out of those models. So that's why if you have very diverse input, then it's hard to model something in the end because you have missing data points or gaps. And that is something I think it's also even more interesting in the pharmaceutical industry, where each pharmaceutical company has also their own standard and the standard operating procedures. And so that is something, if you have, I do not want to say shit in, shit out, because that is not very scientific, but it's really like if you have very diverse data set and a lot of gaps, so then you cannot really rely on the output. That's why it's very helpful if like a community like this CDISC has data standards to put into the community, and this is highly regulated. I hope also for the FDA and for the EMEA and also for the Asian regulatory authorities, so that they have all the same standards, so that we do not make the trial in the US and then in Europe and then in China. So that would be nice if we all apply to the same standards and regulations and do not do very different stuff. But maybe that's also a good insight which we can ask Priya. So for that, if she has anything to tell about these, at first, the data standards, and then maybe I can then after that talk about the literature, which is also maybe something of interest for this community here.
Yeah, no, I think the only thing I would add on to that, Robert, is I think that, you know, some of these data warehouses that we're sort of generating now with novel unstructured data sources, it's really testing our ability to utilize that data uniformly. And I think we need to, as an organization, as a society, as a field, come together in terms of developing some of those best practices and make that well known to everyone so that they can be followed and so it's uniform across the board. Because otherwise we do end up having instances where it's difficult to translate the outcomes and the research from one publication to another. And that impacts science and essentially slows down science. So I do think, yeah, that's the only other sort of aspect I would add is the advocate for development of best practices.
Yeah, that's perfect. Yeah.
If we look at, you know, for example, the pandemic that we had and the accelerated output of research and the rush to market for certain cures. I mean, how can you ramp up something like model MID3 to come to serve in a crisis? I mean, can that even be replicated? Can we even apply it? Or
absolutely, I think that's certainly something that's being done across the board, because not only are we impacted in terms of the need to develop therapeutics really quickly, but also by the fact that resources are limited, as we're seeing, you know, supply chains be essentially affected across the course of the pandemic. So I think, yeah, it's certainly MID3 saves time. It streamlines drug development. It essentially accelerates drug development in so many ways. And a lot of the ways in which we can do that is by just leveraging all of the data that exists before us, right? Using all the different sources of data and all the types of modeling, for example, that I went through to answer questions that are relevant to drug development and regulatory bodies in a very quick and efficient manner. And MID3 has been doing that for a very, very long time, and to the point where we are able to create clinical trials that are smaller in size, that are existing over a shorter time frame, and sometimes even reducing the need for an additional trial, because if a clinical trial if the answer to a question that's being posed by a clinical trial can be answered with the data that already exists, then we essentially eliminate the need for additional trials. So every one of these types of models that we build in MID3 really helps to increase that probability of success, that to result in the selection of the right dose for each patient, and that it really accelerates drug development, and it's something that we've seen across the board for the past 2 decades or more.
Maybe Priya can answer this. So in the COVID pandemic, so I think most of these vaccines were also trialed on younger people, but the most effective stuff was going on in the older population. So can we take this also for a or is it mandatory to hand in like these models for if you have not the population, the target population you cannot reach, because if you make any trials on the 70 plus or 80 plus population, so they will have so many side effects because they are taking so many other drugs like concomitant medication. So does that also add to the drug submission? So is that
Absolutely.
something pharmaceutical industry is doing then?
Yeah, I think that's a really great question. So, you know, I think what we do when we build these quantitative frameworks is we can leverage them in order to bridge between populations. And I think that's really what you're getting at here, right? How do we bridge to a geriatric population? How do we bridge to a pediatric population, to pregnant women, et cetera? And I think population PK modeling, PK modeling, that physiologically based pharmacokinetic modeling that I was referring to, and exposure response or exposure matching allows us to be able to essentially identify doses to be tested in those special population efficacy and safety trials. And then we can go back and confirm when the trials are run that in fact those were the appropriate doses to be selected. So MID3, the quantitative framework we develop allows us to be able to bridge between those populations that are a little bit harder for us to either recruit into or individuals that have, like you said, many other comorbidities and concomitant medications that could potentially result in drug-drug interactions, et cetera.
And Natia posted in the chat, I'll just read it. when I see an M&S report, it's not always clear what variables, known and unknown, and how they have been considered in building the model. Also, the assumptions that were made are not always clear. Are there standards for reporting these in an easily understandable manner?
Yeah, that's a really great question. Thank you, Nithya, for asking that question. You know, I think it depends sort of on what modeling and simulation report you're looking at. And I'd love to know more about that, because in drug development in industry, certainly, there are sort of standards that we tend to follow in terms of how we report things. Certainly PK is something that we've kind of all, I think, across the board standardized in terms of the variables that we are looking at, the parameters that we're trying to estimate, and the interpretation of the outcome. But when it comes to pharmacodynamics models, when it comes to QSP models, when it comes to PBPK models, it's a little bit more challenging to do so, because there are a lot more variables that are being used. When we look at things like QSP and PBPK modeling, we're suddenly looking at physiological parameters. And at that point, really, it's the parameters can grow in size depending on what it is that a person is trying to characterize. And it gets really confusing at that time, I know, to read. But the idea there really is to break it down and kind of understand how do each of these different parameters contribute to our overall understanding of that disease, for example. So I think in short, really, the answer is a good report that's written will clearly outline the assumptions that are made, and also the inputs and outputs. But I do think that it's not uniform depending on the type of modeling that you're looking at.
Thank you.
Certainly.
Now, if anyone else has any questions for our expert panelists, now's the time. We're reaching the end of our webinar, so feel free to ask any questions. We also have one more poll to surface. Gabi, if you wouldn't mind loading that up. We just want to gauge your level of understanding now at the end of the webinar. After this presentation, you know, choose, do you feel more informed or knowledgeable, or are you planning to leverage this information in any way? And while people are answering that, Priya and Robert, do you have anything else that you'd like to add as we near the end of our webinar? I think we're going to run a little bit. We're going to give you back a few minutes time today, because our third panelist Vikram was not in fact able to join us, so we have a little bit of extra time. So we'd rather just give that back to you rather than just keep you on the line.
I don't necessarily have anything else to add, Sharon. Just thank you for the opportunity to come and speak today. And hopefully people have walked away with a deeper understanding of MID3. It's really been a pleasure to be here. And also, if anyone has any questions related to PSP, feel free to reach out to the editors in training, myself, as well as my colleague, Rajith Desikan, who's not here with me today, but we'd be happy to help answer any questions we can about the journal. And we welcome, submissions and things like that from the great work that everyone's doing in the community.
Thank you so much, and we were thrilled to have you. Thank you so much for your time and your expertise. Robert, you as well. Thank you for your time and expertise. It's been great. And on behalf of Research Solutions, Wiley, and the American Society for Clinical Pharmacology and Therapeutics, we thank you for taking time to listen to us today and to learn more about MID3. Now let's see, do we have the poll response? Interested to know people's knowledge level or their intentions with the knowledge they've gained today. Okay, so well over half of you are more informed. Hopefully it was that first half that hadn't been exposed to it yet. Yeah, maybe a few in the other group. You also feel more knowledgeable about how to achieve more predictable results. That's good, about 1/5 of you. And you're planning to employ some of the insights in what you've gained. And I'm happy to hear that none of you thought that this doesn't apply to your line of work. So this is very, this was a very focused webinar on some, key aspects of research. So we're glad that we could have been of service today to share this information with you. And you're also interested in attending other webinars related to this topic. So we'll get to work on that for you. So please look forward to other webinars in this thought leadership series sponsored by Research Solutions. And thank you everyone for your time tonight and for your attention.